I want to talk a little bit about what's known as neural plasticity. And neural plasticity is an absolutely essential function of some neurons to alter their connectivity over time. And that can occur through a couple different mechanisms. One is that neurons can grow and actually form new synapses or more synapses than previously or currently exist, creating greater synaptic connections between certain cells. And that, of course, is a relatively slow process, but it's very important to the development of our nervous system over time. Another mechanism of modifying neural connectivity is altering synapses that already exist. And in that scenario, that really boils down to altering the expression of proteins involved in the functioning of those synapses. And what we'd like to look at right now is an example of that called long-term potentiation, right? Abbreviated LTP. Now, there is sort of an opposing or an oppositely directed change called long-term depression, which is essentially the opposite. So if we look at one, we'll kind of understand the other. Now, in long-term potentiation, and this is thought to be a mediating mechanism for storing memories and learning over time, right? Generating these more complex cognitive functions. And in this action, in this long-term potentiation, what happens is activity at particular synapses results in greater connectivity. In other words, stronger connections at those same synapses. And so in our example here, we've got a presynaptic cell here firing a nerve impulse, increasing our calcium influx. We're going to release our neurotransmitter in response to that. And our neurotransmitter is going to bind to a couple different receptor types over here. Let's start at the top. The first important receptor type is this NMDA receptor. And it's a very strange receptor because it's both ligand and voltage gated, right? So it actually requires multiple stimuli to open it. But having said that, once our neurotransmitter binds and these other elements occur, we're going to get calcium influx. And that calcium influx is going to lead to, through a variety of mechanisms here, either more channels or mediating directly through a second messenger, stimulation of this other receptor known as the AMPA receptor, right? So we're going to end up with more channels, and we're going to open this channel, and now we're going to get sodium influx, right? So we get depolarizing effect of this along this pathway. Right? And now what this means, what's happened here, is that the opening of the first channel not only opens the second channel, but because it activates protein synthesis to produce more of those channels over time, what that means is that release of neurotransmitter at this synapse will effectively result in larger and larger, in this case, because it's sodium, excitatory input to this postsynaptic cell, essentially increasing the connectivity, meaning that a stimulus from a presynaptic cell generates a larger response in the postsynaptic cell. Now, there's one other mechanism we want to point out here, and that involves a channel found in the presynaptic membrane, and that's this kainate receptor and channel. Right? And this guy is actually stimulated by the neurotransmitter to result in ion flux that results in greater neurotransmitter release. So this guy literally forms a positive feedback loop, right? Because neurotransmitter, right? We get an action potential in this presynaptic neuron, we release neurotransmitter, that goes off to the postsynaptic cell, but it also feeds back to stimulate the presynaptic cell to release more neurotransmitter, which of course activates the postsynaptic cell more and releases more neurotransmitter from the presynaptic cell, and so on. And so again, increasing our connectivity between the pre and the postsynaptic cell in this form of neural plasticity.